Hello everyone, my name is Gerry and I'm one of the technical application scientists for BioLegend in the Australia and New Zealand region. I'd firstly like to thank the ADNF committee for organising this event and giving BioLegend the opportunity to sponsor. I think this is a wonderful initiative and I'm very much looking forward to all the talks today. So BioLegend is a privately owned global biotech company incorporated in 2002. We're headquartered in San Diego, California, and this is a picture of our new headquarters, which opened in mid-2019, along with a picture of our founder and CEO, Jean Lay. Our mission is to enable legendary discovery from research to cure, and we aim to fulfill this mission by developing technologies and manufacturing products across research areas, including but not limited to neuroscience, stem cells, immunology, proteogenomics, and oncology. Today, I just want to highlight some of our neuroscience applications. We provide solutions for isolating your cells of interest with MojoSort, which can be used with magnetic columns or tubes for magnetic cell separation, as well as our huge range of flow cytometry antibodies and reagents that some of you undoubtedly know us for. We also make a range of recombinant proteins and antibodies for proliferating and differentiating cells. These include reagent grade and GMP grade growth factors, cytokines, and biofunctional antibodies. In terms of analytical assays for measuring biomarkers, we have common and unique ELISA kits, as well as our multiplex assay LegendPlex. For visualizing your samples, we have a large catalog of antibodies for staining, including neuronal, glial, and microglial markers, and a number of unique antibody clones. Finally, to characterize cells, we have our multiomics sequencing platform, TotalSeq, and our range of Western-ready antibodies and reagents. I would like to just talk a little bit more about TotalSeq and single-cell multiomic profiling in the last few minutes of my talk, as it has really come into its stride in the last few years and was named Nature Methods of the Year in 2019. So our TotalSeq antibodies play a very important role in single-cell multiomics, as they allow for the ability to simultaneously profile and multiple cellular compartments. They do this by integrating into the single-cell RNA sequencing workflow and allowing for the simultaneous profiling of protein and RNA expression in single cells with a method called CITE-seq. So unlike a traditional fluorophore tag that gives a fluorescent readout and where multiplexing is limited by the number of different emission spectra that can be distinguished from one another, the oligonucleotide tail on TotalSeq antibodies instead gives us a sequencing readout, which has essentially unlimited multiplexing capacity. So in addition to a sequencing compatible PCR handle, TotalSeq antibodies have a 15 base pair antibody barcode that is unique to each antibody. So every time a particular barcode is being detected in the sequencing, it means that the protein this particular antibody is binding to is also being detected in the sample. Finally, the oligonucleotide has a tail that mimics the poly-A tail present on all naturally occurring mRNA transcripts. This lets the single-cell RNA sequencing workflow to recognize TotalSeq antibodies as a transcript, allowing for the sequencing readout. Since every unique antibody has its own unique barcode that can be identified bioinformatically, this allows you to combine really as many different antibodies as you want into one experiment, with recent studies using hundreds of antibodies in their panels. So compared with traditional methods, for example, immunohistochemistry, that usually only allows for identification of cells based on a few proteins, multiomic methods gives us the ability to phenotype and identify single cells in a much more detailed manner. And I just want to highlight how far these technologies have come, with TotalSeq antibodies making the development possible. So building on the CITE-seq method, which allows for simultaneous profiling of proteins and the transcriptome, a number of new multiomic technologies use TotalSeq antibodies to simultaneously profile multiple different cellular compartments at the single cell level, and a lot of you may be familiar with some of them already. For example, by integrating TotalSeq antibodies into the single cell ATAC-seq workflow, we can profile chromatin accessibility and proteins with ASAP-seq. And also a recent publication outlined the trimodal profiling of chromatin accessibility, RNA, and protein compartments with a method called T-seq. Further, by integrating TotalSeq antibodies with spatial transcriptomics, another recent publication showed the new method blitz that allows for simultaneous profiling of proteins and transcripts in a spatially resolved manner that also integrates in with single-cell RNA sequencing. Finally, BioLegend has also partnered recently with Mission Bio to release the first platform for simultaneous single-cell profiling of DNA and protein. So all of these multiomic technologies that utilize TotalSeq antibodies, and particularly the spatial technologies, are extremely relevant for facilitating neuroscience research. For example, being able to perform multiomic profiling of single cells in context throughout development is, in my opinion, extremely exciting. So thank you very much for your time. I'd also like to mention that BioLegend is always interested in forming partnerships with researchers. As an example, we work actively with groups at the Garvan Institutes and also other institutes around the world to test emerging technologies. So if there are any technologies that you'd like to work with us on, or if you would like to discuss anything I talked about today in more detail, please get in touch with me or my colleague Hong Yan, or you can check out our website. I've also listed the contact details of our distributors in Australia and New Zealand, and I will stay around in the chat, so please feel free to message me there. Thank you very much.